This video, I am Dr. A. Marmugam, Assistant Professor and Head, PG and Research Department of Botany, JJ College of Arts and Science, Autonomous, Pudukkottai, Tamil Nadu, India. In this video, I would like to emphasize a few points about the ribosomes. The ribosomes, the word derived from the ribo, from the nucleic acids, and some from the Greek word is soma, which means body. So ribosomes are tiny, spheroidal, dense particle about 150 to 200 angstrom in diameter. That they are primarily found in most of the prokaryote as well as the eukaryotic cells. They are the site of protein synthesis. They are the structures containing the approximately equal amount of RNA and the proteins and serve as a scaffold for the order interaction of these numerous molecules involved in the prokaryotic synthesis. Next, the ribosomes occurs in the cell, both eukaryotic as well as the prokaryotic cells. In the prokaryotic cells, the ribosomes often occur freely in that cytoplasm. But in the case of eukaryotic cell, the ribosomes either occur freely or attached with the endoplasmic reticulum. The location of the ribosomes in a cell determines what kind of protein it makes. What kind of protein it going to make it is like based on the ribosomes, it will be the cell will be determined. So the ribosomes are the floating freely throughout the cell. It will be making the protein that will be utilized within the cell itself. When the ribosomes are attached to the endoplasmic reticulum, it refers to as a rough endoplasmic reticulum. It also involved in the synthesis of lipids, protein as well as some steroids also. The proteins made on the rough endoplasmic reticulum are used for the usage inside or cell or outside the cell. Then the average number of ribosomes in a mammalian, there are about 10 million ribosomes are it concerned. Next, the Palade was the person who studied about that ribosome first in the year 1955. And the types of the ribosome is concerned. One is these are the protein decides domain by cells. The plasma membranes that makes the protein for the transport outsides. So the types of it is a matrix ribosome. So first it is a matrix. When it is scattered or embedded in the cytoplasm, it is a matrix ribosomes. The synthesis of protein describes to remind the cell. And this in this image you can see here the ribosomes are available, this in the here also available. Next, the structure of the ribosome. A ribosome is made up of the complex of RNA and protein and is therefore a ribonucleic protein. So the protein which is synthesized from this ribosome, it is a ribonucleoprotein. It around the 37 to 62% of RNA is comprises of RNA and the rest of this protein. So the RNA is maximum comprised with the ribosomes also. Then each ribosome is divided into two subunits. One is smaller and another one is larger. The smaller is which is bind with the larger subunits and the mRNA pattern, and the larger subunit is which bind with the tRNA, the amino acids, and other the smaller subunits also. The prokaryotic have the 70 ribosomes respectively. The subunits comprises of little subunits of 30 As and the bigger subunit of 50 As. There is a small subunit called a 16 S RNA subunits consisting of 1540 nucleotides bound in to 21 proteins. Only it bounded with 21 proteins. The largest subunits composed of 5S RNA subunits, that is 120 nucleotides. There is 23S RNA subunits comprises 2900 nucleotides and in the 31 proteins. The eukaryotic have 80S ribosomes respectively. The comprises of little 40S and substantial with 60S ribosomes. Next, the smaller 40S ribosome subunits is prolate, the ellipsoid and shape and consist of one molecule. 15S ribosomes RNA. and the protein number is 60. The largest 60 is ribosomal subunit is around the shape and containing a channel through which the growing the polypeptide chain it makes its existence. And it consists of three types of RNA. One is rRNA, 20 is rRNA, 5.8 rRNA, and 5 is RNA, and 40 proteins, namely like L1, L2, L3, and so on. So this is structure is representing the ribosome, which is binding with the mRNA, how it is transcribing, how it is making the message from the genetic DNA, like that. So the differences between the ribosome of bacteria and the eukaryotic are used to create the antibiotic that can destroy the bacterial infections without harming the human cells. The ribosomes seen in the chloroplast of mitochondrial of eukaryotics are comprises of big as well as little subunits composed of protein inside 70 is particles. The ribosomes share a core structure that is similar to all ribosomes despite the difference in its size also. Then the two subunits fit together. The work as one translate and mRNA into the polypeptide chain during this protein synthesis also. Because of the formed from the two subunits of non-equal sizes, they are called as diameter. During the protein synthesis, when multiple ribosomes are attached with the mRNA strands, this structure is known as called polysomes, and the existence of the ribosome is very temporary. After the synthesis of polypeptide, the two subunits separates and they are reused or break up upon. Then what are the functions of the ribosome means? The first, the ribosome is a complex molecule, a machine bound within a living cell that serve as a site of biological protein synthesis. This is translation. And another one is the ribosome link amino acids together in order to specific by messenger RNA, that is mRNA molecules, and the ribosome act as a catalyst in two extremely important biological process called as a peptide transfer and the peptide hydrolysis. Thank you very much. Thank you for watching my video.